Welcome to this webinar of the Jerusalem Press Club, JPC, another one in a series, Israel is Alive and Kicking. This morning with Professor Naomi Habib of the Hebrew U, who will speak on handy, simple, and cheap testing kits developed by her team. Professor Habib is a member of the Edmond and Lily Safra Center for Brain Sciences at the Hebrew University of Jerusalem. Her research focuses on trying to solve the secret of how interactions between different cell types in the brain are contributing to cognitive decline and to cognitive resilience, in particular in the context of aging-related pathologies. I hope I managed to give a fair description of what you're doing. But the coronavirus crisis made you, like many other scientists and researchers, switch to a more urgent task. So please tell us about it.
Okay, thank you very much for this kind introduction. So that's indeed true. In normal times, I study the brain, and right now I partnership with Professor Nir Friedman at the Hebrew University, who also at kind of normal time studies yeast genetics, he studies blood cells, so apparently unrelated to the COVID-19 crisis. But what we did is we said pretty early on, we identified that the most critical need for us as a country and in the world to get out of this crisis is for massive testing of individuals that will enable us to go back to our routines while having massive massive testing that will be able to identify carriers of the disease, outbreaks, regions of outbreaks, etc. And unlike the current policy, which is doing kind of more a minimal testing of only people that already have symptoms, and that's really what requires us to stay on quarantine at home. So how are we actually able to help? It's because of the technology we're using on a daily basis. So we usually study a molecule called RNA, which I used in order to study brain aging and Alzheimer's disease. But in fact, if you look at what our diagnostic tests made of, they're all based on the fact that we're identifying these small RNA molecules of the virus, capturing them very efficiently, and then we have a diagnostic test that says for every individual, could we detect this viral RNA, yes or no? So in fact, we have a expertise finding these rare RNA molecules, detecting them in atoms. So we said, why won't we try and make this process much more efficient at scale? So the first thing we did was to look at the current pipeline of diagnosis that's used throughout Israel and identify the bottleneck. So the bottleneck in this process was once the samples arrive to the hospital, these RNA molecules need to be extracted and separated from a lot of other mess that's being gathered with it. And then the second process, putting this RNA into a machine that can detect the viral RNA. And the bottleneck was actually this part of extracting the RNA from the rest of the swab test and all the other mess that comes with it. So what we did is we used an alternative way to do exactly this step. So it integrates perfectly into the current pipeline, diagnostic pipeline that's been used throughout Israel and throughout the world. And what we did is instead of doing a complicated RNA extraction process that involves commercial kits, we actually replaced it with a homemade version of from using almost only materials that can be produced in every simple biological lab. And it's all based on tiny magnetic beads, microscopic beads that can efficiently capture the RNA and that's why our method is currently much more efficient, up to 10 times more efficient than current RNA extraction methods. And most importantly, it's much cheaper, and it uses reagents we actually have in-house. So all these bottlenecks of the missing reagents that kind of stop testing, and also the bottleneck of the most time-consuming step within this process is actually solved by our very simple technology that's now freely available. We already published our protocols online, and we've partnershiped with Hadassah Hospital, with Professor Dana Wolf and Professor Yuval Dor from the Hebrew University, that already have established a very tight collaboration between the Hebrew University and the Hadassah Hospital, where the Hebrew University is actually helping Hadassah conduct testing as volunteers. And so we could come with our technology and to test it, and we could show that over hundreds of samples, our method is similar to the current methods.
It's been said that your invention or development or the kit is 4 to 10 times faster than the current one. Can you explain how it works?
Sure, yeah. So it's actually the tiny magnetic beads, their quality is that they can very efficiently bind to molecules like RNA. So when we put the magnetic beads into the swab test, we can actually capture all the RNAs that is there, but because they're magnetic, we can then use a magnet to capture the bead, just very easily extract all the other materials that comes with the swab, all kinds of proteins and detergents and a lot of other materials. And then we use a very nice trick that when we change the liquid that surrounds the beads, we can actually make the RNA be released from the beads and go into the media. So by using this small trick, we can reduce the whole processing time from several hours to 30 minutes. And that's why it's much more efficient. But to be honest, we're not stopping there, because just replacing the pipeline is not actually the goal we set. What we set was to actually have really long-term efficient testing. And that's why we're currently developing the next method, which is not going to be based on the current method that used RT-qPCR, but rather using genomic sequencing technologies. So will enable us to do 15,000 samples at once and sequence them, so we could really do this large testing throughout the population.
Do you also use robotic activity here?
Yeah, so that's actually a key in our technology, that our protocol works very nicely on a robotic system. So right now it works on a robot that has it can do 96 samples at once. So it means almost 100 samples within 30 minutes. And that's really important for scaling up. And it's also important because it means it can be easily operated by many systems if they set up the robot.
You teamed with Professor Nir Friedman of the Institute of Life Sciences and School of Engineering and Computer Sciences. Is this a time where researchers and scientists come out of their labs, but they're isolated usually, and they team together because only teamwork can come up with these kind of innovative solutions?
Yeah, that's a great question. So actually, science always works through collaboration. So we routinely team up together to solve problems coming from different approaches. Together really kind of sparks innovations. But I think what's unique about this time is we're going out of our academic area. We're teaming up with hospitals much more. We're talking to governments right now, because I really think the solution to the crisis needs to involve scientists that come with the new technologies, comes with the innovation. We don't have any political agenda. Our only agenda is to help the world kind of get out of this crisis. So I think we should really, and I see that also with my collaborators that I'm talking to around the world, in Europe, in the United States, in other countries, that everybody has really kind of putting themselves forward, working night and day to enable the technology that will help us get out of this crisis. And we really hope kind of governments will kind of see this and team up with us to make it happen.
The current tests have a very high rate of, I don't know if very high, but high rate of false positives. Can you explain why, and do your tests solve that problem?
So I think our current test is actually maintains the same abilities that the technology has today. The new technology we're developing, the sequencing-based technology, that will definitely reduce a lot these false positives. So let me explain, where do these false positives mainly come from? So when you have, at the end of the test, you actually get a number. So if there is a whole range of numbers. So usually if somebody has a lot of viral material, there will be no false negatives and no false positives. It will be, the result will be very clear. And if somebody has a zero amount of virus, then there'll be actually no a problem usually. The problem comes that this number kind of goes up and up and up until at some point it's not significant anymore. And this is a matter of setting a threshold. So we always have that in science. But there is a part where this is like a borderline threshold. And the current method only knows to give you this number and tell you if was there any indication of this viral gene or not. If we're going from this qPCR-based method to a sequencing-based method, you have much, much more information because you're actually reading the exact sequence of the virus. And that's why we believe the false positives will be dramatically reduced. There is a way to actually handle these false positives already. I think a lot of public health policies do not want to do that, but it's something that as scientists we do routinely, that we determine a threshold where there are borderline results, where you can't say clearly if it's a yes or a no, and then you have to go back to the same people and sample them again. And this could be because somebody has a little bit of noise in the test, or somebody has just started to develop the virus and he had so little amounts that you couldn't say for sure if he's sick or healthy.
So just to make it clear, until you go into the sequencing method, your invention or development only makes the current system work faster and cheaper.
That's true. We only make it faster and cheaper, and it doesn't come on the expense of our sensitivity, but it doesn't increase our sensitivity.
Okay, there's a question here, whether the fact that we are not doing enough tests is due to lack of the appropriate method, or the different components, or due to a specific approach, maybe a tunnel vision regarding what the system thinks should be done.
Yeah, that's an important test. I think it's a combination of several issues. The first, that was really a huge bottleneck in Israel, and I know it also from my colleagues in Boston, for example, it was a huge bottleneck for them for a long time, was the lack of reagents. These commercial kits that were bought overseas that needed to come into our country and they were missing worldwide. People were kind of almost buying them in the black market. There's all these stories. I'm not involved, but I read these stories on the news. So that, for sure, our technology actually completely overcomes this need, and it also will overcome it for many. If we need to continue testing for 2 more years, I think that's critical, that we have something that we can make ourselves that's dramatically cheaper. But the second point is our capacity. How many samples can we do a day? And that here there are 2 components. How fast can we gather samples? So that really requires, I think there shouldn't be any limits to it, but it does require the commitment from the health system to gather the samples in a fast enough pace. And the second is to have an efficient enough method that could enable us to process thousands and thousands of samples a day. So here, the technology, again, actually does help, because the more efficient our method is, the more we can do. But the current technological technological jump we made is really amazing, and it could be 10 times faster, but we really think that's not enough, and we should scale up to, specifically in Israel, to 100,000 samples a day, and not 10 or 20 or 30, the numbers that people are talking right now.
It's been published, I think, 10 days ago, something like that. What kind of reactions do you get?
That's actually very heartwarming. We get an enormous amount of responses from around the world. It's either colleagues, scientists from other countries who are trying to help their own countries get out of the crisis. And it's a lot of more governmental official people that are reaching out from governments, especially from more, let's say, Latin American countries. I think it's countries that are exactly facing the same place we were, with a lack of commercial kit and availability, and the cost is a much higher burden. But also, in addition, we got we get a lot of contacts from commercial companies who want to make it commercial. So it's typical, free for everybody to use. It's not a product that we can sell to anybody. It's not our purpose.
What is your estimate about the timeline for both the faster test and also the sequencing test to become commercial?
So I'm not sure, I can't talk about the commercial aspect, but I can talk about the availability of anybody who actually has a basic basic knowledge in running these biological tests can just download our protocol and run it already. We expect the sequencing, we hope that our next development will be available also within a few weeks, but the testing there and the approval is a whole different process because it's in an orthogonal method. So hopefully it will really make a difference within a short time.
You've been researching Alzheimer's for quite a while, and you took an innovative approach of, instead of the traditional research on neuronal causes, you said, why don't we take a more general look at the cells and see what happens there. You think the way for a breakthrough in finding a cure or vaccine for the coronavirus should come out of this kind of a fresh thinking about the whole problem?
Yeah, that's a really good point. So actually my grandmother used to always say that necessity is the mother of invention. And I really see that the COVID-19 crisis has sparked an innovation throughout the academic community. There's actually, I think, at least 60 groups in the Hebrew University alone that are all studying COVID-19 from the antibody parts, looking for cure, looking for a more efficient way to prevent infections. It's any idea you might actually think of. You can see that even only within our university, and it's true throughout the world. But I really think that in order to find a solution, we can't just continue with the most traditional way we're doing things. We have to think out of the box and think more globally about this is not a unique situation we've been in before. So we can't apply tools we used in previous crises. We have to think out of the box and develop something new. So similar to our sequencing technology that we suggest, I'm sure there are other labs in the world suggesting other innovative technologies. But I think one of these approaches will really take over and make sure we can go back to our routine, even though the virus might actually stay for a few more years.
This search of solutions in time of crisis require that the thinkers and the researchers team up with practitioners, people who really know. And you yourself, you've been, I think, an ICU nurse in St. John's Hospital way back in Arizona. Not true?
No, that's unfortunately that's not something I was fortunate enough to do.
Okay, because I read it somewhere in the papers.
Oh, really?
So,
So don't believe
so
what you read
fake
in the
news, fake news.
fake
But
news.
there's a need for practitioners to tell or to guide or to help the thinkers come up with the really practical solutions.
Indeed. And we actually, our partnership with Professor Dana Wolf from Hadassah Hospital has been critical for throughout this process. So she, by talking to her, we could really focus on understanding what is the bottleneck and also what's possible. So you can develop a technology that nobody can then use that won't be helpful. We have to develop technologies that can easily apply to the system with their own limits. Right? The way that people gather samples, the way that people can then later do that. We won't be the ones who will, at the end, run the whole pipeline. So we need to make sure the people who run it can actually do it. So we're not wasting time on developing technologies who won't be useful. And here, Dana has been amazing, and her input has been really valuable to us.
You mentioned the interest from Latin America before in this development. Can you elaborate on that and maybe explain why this special interest from Latin America?
I can only speculate actually on that. But what I see is that when, and it's true also in Israel, when people are so busy trying to do to maintain their routine. So people who are already running tests don't even have the time to kind of pick up their heads and think of alternatives. But when do they stop and actually do it is when they hit the wall. So when they, for example, they run out of reagents or they cannot afford buying them, then it's the time when they actually stop and look for alternatives. So I think that people who are actually coming to us are either scientists who are looking around and thinking out of the box, or people who are right now in a huge need of an alternative to what current commercial kits are offering them. So my hypothesis is that, and also by what I hear from people who are writing me and explaining their situations in various countries in Latin America, I understand that they're in a real need of an alternative method that would be much cheaper and available to them. And that's why I think we get many more approaches from them compared to other countries.
Can I dare ask you for your opinion about how this crisis is managed in a general perspective?
To be honest, I feel I have a lot of missing variables in the information I have about this crisis. For example, to what extent is this virus contagious? Are children contagious? Are they not? Can they spread the virus? Can they not spread the virus? So I think they are handling a situation where we're trying to define a policy when we have a lot of variables we don't know yet. But what is very clear to me, and that we can learn from other countries who have been managing their crisis differently, is that there is another way to deal with this crisis. And I think the key here is large-scale testing. So if we do these large-scale and for that, we don't actually need to even know these missing variables. Because if we're doing these large-scale testing and we're identifying people before they're symptomatic, and we're identifying regions where there are more infections than others, then we can really kind of open up the closure, enable the economy to breathe, people to breathe a bit more, and just close specific regions that are more at risk. That's just my opinion from reading and what I know so far about the virus.
May I suggest that journalists who wish to follow up on this can contact you for more clarifications or details?
Sure, I'm available by email, and Professor Nir Friedman as well.
Okay, so Professor Naomi Habib, thank you very much for this very illuminating talk. And congratulations on what you have accomplished up till now, and good luck with the next fresh thinking. And stay tuned to our next webinar at 4 p.m. when we have Yaakov Nagel, former head of the National Security Council, who will speak not only about the management of the crisis, but also on the strategic challenges which don't go away, namely Iran and Hezbollah. Thank you all for being with us. See you later. Bye-bye.
Thank you very much.
תודה רבה, היה מצוין.